Quantitative PCR, real-time PCR, or qPCR as it is known. It's similar to traditional PCR in that you require primers for the target region you wish to amplify. As with traditional PCR, you are aiming to make multiple copies of the region of interest via the designed primers. Now let's step it right back. Where do you get the primers to design for this real-time PCR you wish to do? You can obtain the sequence of the gene of interest via sites such as the National Center for Biotechnology Information. And once you have this sequence, you can identify the region you wish to amplify. A reminder that the primers are designed so that you make numerous copies of that specific region relative to the rest of the other genes in the chromosomes of the genome you're studying or investigating. The primers you design flank the forward and reverse region that you are interested in. With traditional PCR, you get a result of whether the region you targeted was present in the genome right at the end of the complete reaction. We'll talk about the number of cycles you generally use for a complete reaction to ensure that you get sufficient copies of the region to be detectable if it is present in the sample. With qPCR, you allow yourself an additional opportunity for gathering information by including either double-stranded DNA binding dyes or a specific fluorescent probe such as TACMan, and this reports to you how much of the region of interest is present at each cycle of the PCR reaction, not just at the end of the complete reaction. So why would you want this kind of information? Well, for certain situations, example viral infections, you don't just want to know whether the virus is present. Sometimes it's critical to know how much of the virus is present in the sample that you're analyzing. In certain diseases, it may be critical in determining whether you actually need to treat or you just observe and monitor the disease to see how it's progressing, because what is considered the viral load, how much of the virus is present, may be what's critical in determining whether you have a problem or you just have an issue that you need to monitor. So with qPCR, what happens with the additional fluorescent molecules or probes that you are incorporating during the PCR reaction is that the fluorescent signal intensity will increase from each cycle of the qPCR reaction if the gene you are targeting is present. This is a result of the doubling of the gene template from cycle to cycle. If we're zooming a little bit at where you're using a specific probe for your qPCR rather than just the double-stranded DNA dye, the probe has a molecule called a quencher that stops the fluorescent molecule from giving a signal while it is not part of the PCR reaction. So the signal only occurs when it is incorporated in the PCR reaction. And so this means that if you are using a specific probe in your qPCR reaction, you design not just the forward and reverse primers, but you also design the fluorescent molecule to bind specifically to a region of the region you are wanting to amplify. And then a quencher is incorporated to regulate the fluorescence while it is not part of that PCR reaction. Essentially, you need 3 primers if you like. Well, the primers, the forward and reverse, and then an additional specific primer that binds to the region. And this specific primer also has a quencher, a molecule that stops the fluorescence being emitted. So as you can appreciate, using this specific probe gives you a layer of specificity, an additional layer of specificity. Now, you don't have to use this specific probe. Some people may just use a double-stranded DNA binding dye. So can you do qPCR on a regular PCR machine? Well, actually no. Because you have the additional fluorescent molecule, you need a PCR machine that has lasers to supply the energy that excites the fluorescent molecules. And then you need detectors to record the fluorescent wavelength emitted in response. So a bit more about these specific fluorescent probes that you can incorporate into your qPCR, or you usually incorporate if you're not using just the double-stranded DNA binding dyes. It is designed to have a molecule such as PHQ at the end of the sequence that stops the molecule from emitting any fluorescent signal when it is not specifically bound to a region that you are amplifying. Now, I keep talking about cycles of the PCR and the fluorescent signal being detected at the end of each cycle rather than at the end of the complete reaction. So let's look at how PCRs in general operate. I probably should give a definition of PCR, polymerase chain reaction. In these experiments, you first denature the double-stranded DNA template that form the genome of the cells or biological samples you are analyzing. The primers bind to the region for which you have designed it to bind on the DNA template. By repeating the denaturation and primer binding cycles, the binding step is called annealing. By doing this multiple times, you copy the region you have designed the primers for. An analogy here is like when you're photocopying and you change the number of copies to however many you need. So how many copies do you need in a PCR? We'll get to that in the next slide, but be aware that the temperature that you need for the annealing step, unlike the temperature that you need for the denaturation, is not set in stone. It's not set in stone, and you will have to adjust and optimize in initial experiments or preliminary experiments to find out. There'll be a bit of playing around, experimenting initially to find out what temperature is ideal for the annealing of your primers. But the temperature for denaturation is generally a set temperature for DNA templates. Now, to how many copies or cycles you need to have sufficient copies of a region if it is present, it turns out that about 25 to 30 copies cycles is generally sufficient, but you may need to go up to 40 cycles in some instances, particularly if there isn't much of the region you are targeting, called copy number. A practical example that we are experiencing in 2020 throughout to 2022 is COVID-19. So with your COVID-19 swabs, for instance, where a person is newly infected and therefore you only have few copies of the virus in the sample, then up to 40 cycles may be needed to detect the regions of the viral genome. Whereas in a person that has been infected for quite some time and the virus has had time to multiply, fewer cycles will be required to detect it. Let's take a look at an amplification plot, a typical amplification plot. You have a lag time where there isn't much or enough of the gene being targeted to give off detectable signal, but with time, with each cycle, the template increases due to the doubling at each cycle. So the cycle where the detection is possible is important in communicating how much of the region you are interested in is present in the sample. An example, if we detect the region of interest after just 15 cycles in one sample, but it took 30 cycles in another sample, then we know that in the sample that had detectable signal after 15 cycles, there is more of the viral gene. In practical terms in the lab, when you're trying to determine how much of a virus is there, you will have a standard curve. You have standards that you use to create a standard curve. So standards are essentially control samples in the sense that you know how much of the virus is present in each point of the standard curve, in each point of the sample that you're using to form a standard curve, because you design it and it helps you quantify how much, for example, if we say the 15 cycle detection, how much this equates to in terms of quantity of virus. So I hope that you appreciate that you're able to comment on how much virus there is only because you have a sample that you have known amounts of virus. And so if you compare your 15 cycle detector with your sample, with your standard where you know how much virus was, then you can make inferences, you can comment on how much virus there will be in a 15 cycle detecting sample. So the way standards work, I do have a video on here about standard curves. It's a critical technique in medical research, in diagnostic laboratories. So take a look at it. So the standards are serially diluted to provide various ranges or points that are used to create a standard curve, a graph of how much virus you expect to have at a given concentration in other instances, but in this case at a given viral load. In addition to the standard curve, in practical terms, you would also include quality control samples. So these are samples where in one, you know for sure that there is virus, and so you expect to detect it. And then in another, you know there is no virus, and you expect to not detect any fluorescent signal. And these help you to trust the results that come off of your qPCR run. The quality control sample gives you confidence in the results from the machine. If the known sample control with virus show up as negative, then you know not to trust the results from that particular run on the machine. They help you validate the run. The samples and the standards that you're running from patients or whatever sources are run as replicates. And this means that not just one instance is analyzed, but multiple instances, maybe duplicates twice or 3 times or more, are analyzed to check that the results are reproducible. So the data analysis side, once you obtain the results, you will need a dedicated software to analyze the cycles at which the detection took place. Aligning the cycles with the amount of virus there is based on the standard curve. The dedicated software will allow you to have a visual of the points on the standard curve. The important thing to watch out for is the reported efficiency of the reaction and the accuracy of the response of the standard curve. The efficiency of the amplification should be around 90 to 110%. The correlation between the amount of virus and the signal response, the R-squared value, should be closely related. So close to 1 is what you're aiming for. So remember, high viral loads show up earlier in the cycles, and this is reported as a CQ or CT value. The quantities may be reported as the scale of the graph, maybe as log values. You can take a look at my video on the use of logarithms if you're unfamiliar with this scale. It's not an everyday scale by any means. And so have a look at it and why logs are the benefits that a log scale offer you. And you must confirm that the quality controls are met before you can trust or communicate the results. It's important to take a look at the shape of the amplification curves to make sure that the amplifications are true amplifications and not just noise or interference. In instances where you need actual quantities for particular diagnostic or disease management decisions, so you're not just interested in the threshold cycle, the CT or CQ, but you actually need numbers, quantities of virus, well then after confirming that the reaction has proceeded as appropriate by the controls, you may determine the viral load you can determine the viral load. This is done by dividing the starting quantity of the viral gene you've detected. This is based off of the standard curve by the starting quantity of a control gene that you've tracked and know that its expression is stable. So your starting quantity of the virus that you're analyzing, you don't know how much there is in the patient, by a control gene that you've also tracked by designing primers for in the same reaction, and so you divide the unknown quantity by the known quantity. Okay, so that's basically how the analysis, the parameters that you have to keep in mind when you're analyzing your qPCR. So now over to you, the person watching this video. Which gene are you detecting in your qPCR assay? I'm going to wish you all the best with your qPCR assay. God bless you. See you next time. Thanks for watching.